everyone. Good afternoon. My name is Haixia Chen, and I come from Tianjin University. I'm from the Department of Natural Products and Traditional Chinese Medicine and School of Pharmaceutical Science and Technology, Tianjin University. This is the contact information. Hope we can collaborate in the future. And this is my biography. Now I'm the professor of Tianjin University, and I used to be the visiting scholar in University of California, Berkeley, and postdoctoral fellow of Ocean University of China, and a PhD in Huazhong Agricultural University. And my research interests mainly focused on the following areas: the natural product chemistry and the bioactivities and mechanism of the constituents from natural resources, and the analysis method study of chemical constituents, and also the functional food and the healthy food. So today my topic is about the isolation and the simultaneous determination of 5 bioactive phenolic compounds in mushroom, Inonotus, obliquus, and evaluation the antioxidant activity. So I will follow the following paths. First, the introduction of the mushroom, Inonotus obliquus. In Chinese, it is called 华鹤孔菌 or 白化绒, and it is always grown on the death birch, just like this photo. And it grows mostly on the latitude 45 degrees to 50 degrees. So it always grows in the cold area, including Russia, Poland, and most of the Baltic countries, and also the northeast of China. This is the taxonomy of Inonotus obliquus. It is a kind of edible and medicinal mushroom. There are some studies have focused on this mushroom, and some chemical constituents have been reported. It mainly included triterpenoids, just like this compound, and the polyphenolic compounds, and also the others such as polysaccharides, amino acids, and proteins, phenolic acids. Because it contains many of compounds, and there are many activities were reported, such as hypoglycemic and platelet aggregation inhibitory activities, antiviral, immune response modifier, antioxidant, antifungal, antitumor, and hyperprotective activities, etc. And also it has been reported on the clinic applications, including the gastrointestinal cancer, cardiovascular disease, stomach ailments, and tuberculosis and diabetes, liver diseases, etc. In the application, especially the diabetes, in Russia, there are some drug companies have produced this kind of drugs. It was reported that in Russia, on the diabetes, the curative rates can reach higher than 19% of the diabetes. So based on the diabetes is the third killer, and it is also the global diabetes will reach very high. It is very promising resources. Also, it can be used as a functional food in the healthcare and antioxidant and immune response modifier. Many researchers have focused their studies on this mushroom. However, about the extraction, isolation, and the structure, especially the structure and activity relationship, hasn't been reported. So the main constituents on the structure-activity relations will be discussed here. So the second part is the isolation of chemical constituents of the Inonotus obliquus mushroom. We use the traditional procedure. First, it is extracted with ethanol and then participated with different solvents with different solvent, so we can get different extracts. Then we screen these extracts on the antioxidant activities. We found that the butanol extracts were the most active one. So we will isolate the constituents of this part. We will use the different kinds of isolate methods, such as absorbent and repeat normal phase silic gel, Safdex LH20, column chromatography, and the reverse phase HPLC, and precise sterilization, etc. Then we get several compounds, and then we use the traditional methods and the modern methods to identify them, such as the physical chemical properties and the NMR and the mass, ESI mass. So we identify them. The first one is the 3,4-dihydroxybenzyl dehydrate, and the second one is the vanillic acid, and the third one is Caffeic acid, and the fourth one is syringic acid, and the fifth one is the 3,4-dihydroxybenzyl. Because the mushroom Inonotus obliquus and its extracts had been used in our life as a functional food or some drugs. So how to control the quality is a problem we should to deal with. So we have developed the simultaneous determination of these 5 bioactive phenolic compounds. This is the HPLC chromatography. So it was found that these 5 phenolic compounds have been isolated and established the method. This is a linear relationship between peak area and the concentration of the 5 compounds, and it was found that all of them have good regression equation with alpha square, and the LOD and LOQ also were also detected. And also the intraday and the interday precision of the developed method was also determined. It was found all of the data were lower than 5%, so it was validated. And also we analyzed the repeatability and stability of the HPLC method, and it was found it was acceptable. This is a recovery data of the developed method, and all the recoveries are between 96 to 102. We try to detect whether our method, so we application, we applicate the developed method, and then we use three extract methods to get three extracts. Then we determine the method and to determine to compare the three extracts. Then we found that the three methods, we get the three different extracts from the same resources, but it was found that the five phenolic compounds contents are different, and the bioactivity, we use DPPH radical scavenging activity assays and MDA assays to detect the three extracts. We found that there is a good relationship between Caffeic acid to the DPPH activities, and the 3,4-dihydroxybenzylation also has good relationship. So it was suggested that these two compounds are the main antioxidant constituents of the extracts. So we come to the conclusion: 5 phenolic compounds were isolated from the mushroom Inonotus obliquus, and a simple RP-HPLC procedure was developed for simultaneous determination of the 5 compounds. And among the 3 extraction methods on Inonotus obliquus, heat reflux extraction was the efficient extraction method with a strong antioxidant properties. And we found that there was stronger linear relationship between DPPH radical scavenging activity and the total phenolic content, Caffeic acid content, and the 3,4-dihydroxy benzylactone content. My interest study on the natural products has been more than 15 years. So I have about 40 SCI papers have been published. This is some of my selected published SCI papers. I'm very glad to be here to share the information, and I hope we can collaborate in the future. Welcome to Tianjin University. So this is the last my acknowledgements. So here I thank my School of Pharmaceutical Science and Technology at Tianjin University provide the equipment and instruments of my study, and all members of my lab. They have hard work, and most of them have published SCI papers during the studies. Thank you. Thank you very much.
Thanks, Dr. Chen. There's one question for me, and then I go for the next speaker. All those 5 compounds you chosen, they are not characteristic. Most of the other compounds also have, right? One characteristic you haven't told us is the polysaccharide. Have you tried to measure quantitatively the polysaccharide?
Yeah, I have studied polysaccharide for many years, and also I have many papers.
Okay, as I said, the 5 compounds are not very specific.
When I study one resource, I will divide them separately, the macromolecules and the small molecules.
Yeah, that mushroom is a very rare mushroom in Russia, in Poland. How much did you collect? By yourself, or by you buy, or both? From where your mushroom originate?
In China, in northeast of China, Chanbai
You collected
mountain.
or you collected?
Not
No.
mine. I just buy it from the company.
I think you should
How
also
much,
collect.
how much did you use your mushroom? How much did you use? Extract or how much?
5 kilometers.
5.
Yeah.
5 kilometers.
Because this mushroom is very big on the beach. But one is very big.
Thank you very much for introduction of the Phytochemistry. This is my boss in France. Okay. Obviously, thank you very much.
Thank you.
You guys can link. Yes.
Yeah, we
Good
can
one.
collect it.